Hello and welcome back to part 3 on the topic of sampling in the series of videos e-learning from Q-Interline. In this video, I will talk about how we present sample to the analyzer, because what does the analyzer actually see? In the video, I will have slightly more focus on at-line analysis, but the question, what did the probe or cell actually see, is equally important for online. When we acquire a spectrum, we want to have a representative spectrum of the entire analytical sample. If we have a very representative sample, but we only see part of it, we may simply lose all the effort we put into creating a representative sample. The good thing is most accessories from Q-Interline will spin and we apply composite sampling. That means we see hundreds of points of the sample. We call it spin and co-add. By spinning and co-adding spectra, we'll get a much more representative sample, but it's not irrelevant how it's done for liquids, pastes, powders, and more heterogeneous produce. So we better watch out for these things. Likewise, it's important that we are aware that a sample may segregate, dry up, sediment, change its temperature, and certain particles may be favored to drop to the bottom of a petri dish and similar stuff. So presenting the sample to the analyzer is not trivial. When we work with NIR, which is the primary expertise of Q-Interline, we have several options. Liquids are typically considered very homogeneous, and they are likewise done in transmission, shining light through them, fairly simple. If that liquid is several phases, it becomes almost impossible to handle with NIR, especially if we have air bubbles, particles, and eventually two liquids that does not want to mix well. That becomes very, very tricky, because what do we actually see? For suspensions, it might be milk. It's a little bit easier because that suspension may be more stable and we may be able to do it in both reflection and transmission. Those are the two sampling modes, and sometimes one is better than the other, but it may depend on the application. Powders, be that fine powders or coarser powders, we have to be very, very certain that we do not create segregation, because what did the camera, the spectrometer, actually see may just be the smaller particles or the bigger particles if we look from above. So we have to be very careful that the spectrometer sees all the particles with the same probability, the sampling theme that we discussed in the earlier videos. With pastes, we want to create a good surface for reflection, or we want to create an equal thickness for transmission. So there are multiple things that we need to consider. For solids, it's a little bit like coarse powders, but for very big particles of solids, it will be primarily reflectance, diffuse reflectance spectroscopy used. I have tried to visualize the sampling challenges in the various accessories, and it's an extract from an article that was brought in Near News, volume 25 in 2014. We used accessories from Q-Interline, but I would say the concerns and challenges are generally applicable. We used 3 accessories. We used the petri dish, the bottle sampler, and the spiral sampler. We produced a binary mixture of polyethylene and polystyrene because they have very, very different spectra. We created mixtures of 2 and 5 and 10 and 20% of these mixtures by weight, which can be done very, very accurately. We corrected for volume so that it's really a true mix volume of these two. We made a calibration, and that calibration is of course quite easy to produce because it requires only 2 components. Let's look into the sampling of a petri dish. The petri dish is easy to use. Powder can be filled in easily, but we see only what is at the bottom. So we better make sure that what's at the bottom is representative of the entire sample. And we don't see the entire bottom. Most of these accessories sees a rim of some 18 square centimeters in area at the side. So the way the pearls, the plastic pellets, fell into the petri dish was very determining. But the good news is it's very repeatable, because if I run another circle, the pellets will not have moved. They will be in the same spot, right or wrong. The next thing is a rotating jar sampler. The intention is to mix and make sure the spectrometer have an equal probability of seeing all the particles. The base area is only 15 square centimeters, but as it tumbles, it will give all the particles an equal chance to come around. The last accessory is a Q-Interline patent from 2014, in which we see a large area of 375 square centimeters. Easy to fill and sees an enormous area. And that will turn out to be the right choice here, hence for binary heterogeneous samples. The experiment was conducted so that we had a base lot, we mixed it, and we did composite sample. We filled 10 of the either petri dish or jars or spirals and scanned them. Then in sampling experiment number 2, we took whatever was in that petri dish, took it out, sampling container, took it out, put it back in. That's sort of like a reproducibility. And then one of those we simply ran 10 times to see the repeatability. So that was the construction of the experiment with the binary thing. The outcome was quite fascinating. Everything was scanned on a quant FTNIR spectrometer. We ran from 4,000 to 12,500 centimeter minus 1. And we ran at a resolution of 16, meaning data point spacing every 8 wave numbers. The spectrum were co-added, time was kept constant, room temperature, and 40% relative humidity. So everything was kept constant. The prediction model simply just involved a small CH region, because the spectra of these two plastics are very easy to model. And with a simple Savitsky-Golay pre-processing, we could make a very nice model. But when it came to prediction, the results were quite different. Let's look a little bit into the results of the first experiment. The results of the prediction, the 3 different sampling scenarios, repeat sampling, repeat filling, and repeat scanning, is shown here in this graph. What we see in this graph is that repeat sampling is quite difficult for the petri and the bottle, whereas the spiral sampler really doesn't care much what portion was taken out, because the area is so big. It is likewise sees that packaging another petri dish, something totally new will fall on the edge, the rim of the petri dish, and we will simply have a complete new result. The only area in which the petri sampler is really shining is on repeatability, because if you do not move the pearls, the plastic pellets, you will see exactly the same. But now, if we look at representativity, we can jump to another plot, which is a plot of where we see, is there a bias? Because you may be very repeatable around the wrong result. So we defined that representativity was bias squared and the SEP squared. When we look at that, it's obvious that for low concentrations of a binary sample at 2% mixture, the odd chance that the petri sampler will actually see 2% of these pearls at the rim is very low, and the representativity is very, very bad. Huge biases are seen. The higher the concentration, the errors drops off, because the chance that there will be a certain amount of plastic pellets in that area is higher. But we also see that by doing a real composite sampling, like what's done in the spiral sampler, we see a huge area chances that we will simply catch much better the 2%, the 10%, and the 20%, but it's also more difficult to repeat because it simply sees something new every time. But in total, seeing a large sample composite sampled is the way forward and will give by far the best results, as can be seen with the green results here. So these are some of the sampling things to consider when you present a sample to an analyzer. That concludes the part 3 of presenting a sample to an analyzer. I hope to see you back in video number 4 on the topic of taking a spectrum. See you there.